focusing on both short and long-term dynamics. So birds are female heterogametic. So males have 2 copies of the Z and females only have 1 copy, along with the female-limited W chromosome. And because of this unequal pattern of inheritance, we predict that the sex chromosomes are subject to unbalanced sex-specific selection and will become sexualized. So the avian Z chromosome is present more often in males than females, and so we might expect it to be subject to strong male-specific selection for dominant male benefit genes. And so typically, when sex chromosomes are sexualized, this has a dominant effect on gene expression. So we might expect an enrichment of male-biased genes on the Z chromosome. And so the question that I wanted to answer today was exactly what the role of this masculinizing selection has in Z chromosome evolution. Is the Z masculinized? And specifically, is this conserved across both short and long-term evolutionary timeframes? So in order to answer this question, the first thing that we need to do is understand how the sex chromosomes evolve. So sex chromosomes evolve from an identical ancestral pair of autosomes, and they differentiate and diverge as recombination is suppressed. So this stepwise cessation of recombination spreads across the sex chromosome over time, and you can call these different regions strata. So in this example, stratum 1 is the oldest and has been isolated from recombination for the longest amount of time. So you'd expect to see the largest degree of mutual divergence between Z and W orthologs. But in contrast, the youngest region is stratum 4, and this is the youngest and therefore the least differentiated. So I was actually able to characterize this evolutionary history of the chicken sex chromosomes using 18 newly identified Z and W orthologs. And so this is mutual divergence between those 18 Z and W orthologs plotted against the physical position on the chicken Z chromosome. And you can see that the orthologs cluster into 4 distinct strata, which suggests that the avian Z chromosome was formed by up to 4 recombination suppression events over the course of around 130 million years. So the oldest strata is here, and the youngest one here at the tip evolved around 40 million years ago. So we can actually use this evolutionary history to examine the selective regime that's driving the evolution of the Z chromosome by comparing patterns of sex-biased expression across these 4 regions. And I was able to do that using transcriptomic data from male and female chickens. So this is average male-biased expression calculated for all genes within each of the 4 strata plotted against the age of the region. So this is the oldest region here and the youngest region here, containing about 40 Z-linked genes. And you can see that over time, male-biased expression accumulates on the Z chromosome. And this is really consistent with the effects of male-specific selection, indicating that at least over long evolutionary time periods, so over the course of around 130 million years, the Z chromosome is masculinized. But what about short-term periods? Is this pattern conserved? And specifically, the questions that I wanted to ask are whether this evolutionary history of the chicken Z chromosome that I showed you earlier is conserved across other avian species, and also to understand how sequence and expression on the Z chromosome evolve over shorter evolutionary time periods. So I framed these questions using a clade of birds called the galloanceriform, which are made up of the landfowl, the galliforms, and the waterfowl, the anceriform. And we have whole genome transcriptomic data for male and female adults for a range of tissues across these 6 species. And they're a perfect system for studying short-term dynamics of Z chromosome evolution, because they diverged relatively recently, so only 90 million years ago, and there's also a huge degree of variation in the degree of sexual dimorphism and levels of sperm competition. So for example, the goose is monogamous, but the duck has a huge degree of sperm competition. So because of this, we might expect different levels of male-specific selection and therefore different extents of Z chromosome masculinization. So the first question that I wanted to ask was whether this evolutionary history of the chicken Z chromosome that I showed you earlier, so these 4 strata, are conserved across the galloanceriform, or whether recombination was suppressed independently in different lineages. So there's no W chromosome assembly for any of the 6 species that I just showed you, but using a combination of bioinformatic approaches with the transcriptomic data that we have, I was able to identify Z and W-linked genes for the turkey and for the duck. And then you can use a phylogenetic approach with these newly identified Z and W orthologs in order to assess when recombination was suppressed. So if it was in the common ancestor of these species or independently in different lineages. And it turns out it's actually a combination of both. So this is a gene tree for a ZW ortholog located in the oldest strata here. And you can see that the duck W and chicken W orthologs cluster together in red, and the Z-linked orthologs cluster together in blue. And this indicates that recombination was suppressed in the common ancestor of these species. And you see exactly the same pattern when you look at genes located in stratum 2, where orthologs cluster together by sex chromosome, so W-linked orthologs in red and Z-linked orthologs in blue, and they don't cluster together by species. And so this really suggests that the oldest two strata evolved in the common ancestor of these species and are conserved across the Galoanceriform. However, you see a very different pattern when you look at the youngest two strata. So this is a gene tree for this ortholog here. And you can see that the duck, W and Z orthologs cluster together independently from the chicken and the turkey. And this obviously is a very different pattern from these two gene trees above. And so this clustering by species and not by sex chromosome indicates that recombination was suppressed independently in these different lineages. And so you can build gene trees for all of these Z and W orthologs, and essentially they show that the oldest two strata are conserved across the Galoanceriform, but that there were two independent strata formation events in the Galliforms, so the lineage leading to the landfowl, and one independent event in the Anceriform, so the lineage leading to the waterfowl. And interestingly, the time at which recombination was suppressed independently in these two lineages is relatively comparable, so about 30 to 40 million years ago. And so now we've established that we have relatively comparable evolutionary histories of the Z chromosome across these clades of birds. We can start to compare gene expression on the Z chromosome across these species to examine how Z-linked gene expression evolves over shorter evolutionary time periods. So we might expect the extent of Z chromosome masculinization to differ across these 6 species, because they vary in the degree of sperm competition and therefore the degree of male-specific selection. So I was able to calculate average male-biased expression across the whole of the Z chromosome for each of the 6 species and plot that against 2 measures or proxies of sperm competition, residual testes size and log sperm number. And I was able to do that for both the gonad and the spleen. And you can see that for both tissues and for both proxies of sperm competition, there's no correlation, and this is after you correct for phylogeny, between Z chromosome masculinization and sperm competition. And so this isn't very consistent with the idea that short-term changes in male-specific selection are driving gene expression evolution on the Z chromosome. So we failed to establish a link between masculinizing selection and short-term changes in Z-linked gene expression evolution. But what about sequence evolution? So the Z chromosome has an effective population size 3/4 that of the autosomes because of its unequal pattern of inheritance. And therefore we might expect drift to act more strongly on Z-linked genes relative to autosomal genes. And in support of this, you do see faster rates of change of Z-linked genes relative to the autosomes. So across this clade of birds, you do see that DNDS on average is significantly higher for Z-linked genes relative to autosomal genes. And you can call this the fast Z effect. However, we might expect this relationship to differ depending on mating system. And this is because as the variance in male reproductive success increases, the effective population size of the Z relative to the autosomes decreases lower than 3/4. And therefore, the strength of drift is predicted to increase. So under increasing skew in male reproductive success, so increasing promiscuity and increasing sperm competition, you'd expect drift to act more strongly on the Z chromosome and you would predict a faster fast Z effect. So we can test this directly using these 6 species and those 2 proxies of sperm competition I mentioned earlier, relative testes size and sperm number. So this is the strength of the fast Z effect calculated for each of the 6 species and plotted against these 2 proxies of sperm competition. So the first thing to notice is that the Z chromosome does evolve more rapidly than the autosomes in each of these 6 species, because the strength of the fast Z effect is greater than 1 in each of the species. And the second thing to notice is that there is a significant correlation, significant positive correlation, and that's after, again, you correct for phylogeny, between the strength of this fast Z effect and these 2 proxies of sperm competition. So this really indicates that as your skew in male reproductive success increases, and we're using sperm competition as a proxy for this, the drift acts more strongly on the Z chromosome, and you see a faster fast Z effect. So we can see that the Z chromosome is evolving with a significant contribution of drift, and ultimately this actually may be acting to constrain gene expression evolution on the Z chromosome by limiting the degree of standing genetic variation. So just to conclude, we've shown that the Z chromosome over long evolutionary time periods, so over the course of around 130 million years, is masculinized. But over short-term time periods, we fail to establish a link between short-term changes in the strength of sex-specific selection and gene expression evolution. And this may be because ultimately there's a trade-off acting on the Z chromosome between the strength of drift and the strength of sex-specific selection that's ultimately acting to limit the role that the Z can play in encoding sexually dimorphic phenotypes. And I'd just like to say thanks to my supervisor, Judith, Mank, and everyone who contributed to this work, and also point out that there are 2 other talks from the group, Becky, who's talking later in this session, and Peter, who talked this morning. Thank you very much for listening. Any questions? Yes?
Yeah, how about positive selection? Is the world just a really sad place where nothing but bad shit happens?
So, no, that's a great question. So that correlation could potentially be because of positive selection. So it could be that in females, the Z chromosome is exposed, and therefore selection can act more efficiently on the Z chromosome. But you can kind of...
Or if there are male favorable genes that are selected in males.
Definitely. Yes. So you can kind of start to disentangle that effect if you look at different expression categories. So I have a slide here that hopefully should explain that. So if you look at different expression categories, if it was, say, because of exposure of the Z chromosome in females, you might expect a stronger fast Z effect in female-biased genes. Or if it was because of positive selection acting on male-biased genes, again, you'd expect to see a kind of faster fast Z effect in male-biased genes. But when you look across these expression categories, there's no significant difference between the strength of the fast Z effect. And there's also, when you do tests of positive selection, there's no enrichment of genes on the Z that are undergoing positive selection. So I think it's probably drift that's driving this now. Yeah, that's a good question.
Another quick question?
And the relationship between the testis size and the fast Z effect?
Yeah.
It's a bit like a kind of group effect. Stuff in circulating something weird. Is there anything about this?
These 2 species here. Not that I can see immediately, but I do agree that potentially these 2 measures might not accurately reflect effective population size 100%. Because, for example, the peacock here, you might expect, because males are dominating mating, that they also have a very kind of skewed variance in male reproductive success, and so a different effective population size of the Z relative to the autosomes. So one of the things that I also want to do is go and actually calculate with polymorphism data effective population size of the Z, and also use that as another measure to kind of test this relationship exactly. But yeah.